And last up, we have Dr. Berenice Benayoun, who's at USC, Southern California, and her title is Sex-Specific Remodeling of Aging Murine Macrophages.
Thank you, Ashley, and really thank the organizers like Peter for putting together this event. I think this has been really exciting, and hopefully I can maintain excitement. So like Ashley said, today I'm going to tell you about some of the work my lab's been doing to understand sex-specific patterns of omic and functional remodeling with aging in the immune system, and here specifically macrophages. And I'm sure you all saw a couple weeks ago an updated Hallmarks of Aging came out, and I was actually really excited to see that it now includes chronic inflammation as one of the hallmarks, because this is really something that we and others have seen being really a recurrent feature across tissues, cell types, species as being really characteristic of aging. And so this has also been sort of highlighted and coined under the term inflammaging by Claudia Franceschi, this idea that with age, and probably due to repeated exposures during the lifespan, there is a chronic activation of the innate immune system leading to this chronic inflammatory state that might in and of itself be deleterious and create some of the phenotypes of aging. And so in my lab, we're really interested in understanding how that happens, but also with a highlight on how sex dimorphism and biological sex plays a role in this. And this is why I like to say that with the way people normally do aging research, we might be missing half of the picture. I think Ashley, Dr. Webb did a great job at the beginning of the session explaining some of the issues in the field with the overwhelming majority of studies that have been conducted in the aging field, especially when there's a molecular angle to it, being done exclusively on male animals. And when you talk to people, there's usually a lot of explanations as to why they chose to do only males, ranging from that it's too expensive to do both, to female data is too noisy, to my favorite, females are just males with hormones, which by the way, they're not. So, you know, if there's one thing you get from my talk today, please let it be that. But because of that mindset, we're in a situation where we know very little about differences in the way females and males age, especially at the molecular level, which is where we can really get insights into potential therapeutics. And so why is that important? Well, it turns out that over the past decade or so, like maybe even more now, we've had a great NIH-supported program, the NIH Interventions Testing Program, to sort of cleanly determine what interventions are going to have an impact on, you know, slowing down aging or at least delaying the health issues linked to aging. And I think what the biggest finding from this consortium has not been that this drug or that drug can delay age-related decline, but really that there's literally not a single intervention that they've tested that works the same in females and males. You have drugs that work exclusively in one sex, or drugs that work much better in one sex than the other, which really suggests, at least to me, that, you know, if aging was exactly the same thing in females and males, then you would expect the same drugs to have the same effect in both. And the fact that they don't means there are some fundamental differences that we need to better understand so we can, you know, design longevity interventions that are tailored to individuals. And there's a lot of other things telling us this is not just going to be a mouse, you know, specific issue. We know that menopause in humans is literally the single biggest risk factor for women to develop all sorts of age-related diseases, including Alzheimer's, for instance. Suggesting that, you know, reproductive function, reproductive hormones, and sex in general will impact the way you age. A big consortium like the NIH Roadmap Epigenomics Consortium showed, and they were really surprised by this, that the biggest difference in every single tissue where you do epigenomic profiling The biggest difference is not the age of the individual, it's not the disease status of the individual, but really whether the tissues come from female or male individuals. And so I told you we're really interested in the immune system. Well, it turns out there's a lot of reasons to believe the immune system itself, regardless of age, is very sex-dimorphic. We know, for instance, that for every man that has an autoimmune condition, you can find 9 women. So suggesting potentially a more active immune system, at least in young women. But conversely, we know that outcomes after infections or sepsis are always much worse in men. So again, the immune system, clearly sex-dimorphic. How is that influenced with aging? And so this is a question that a very talented graduate student in the lab from the Biology of Aging program, Ryan Liu, has been interested in elucidating. And he started really with the question of whether we could identify and understand sex dimorphism in the aging immune system. And so at the time, my lab was very new in the immune domain, and so we wanted something sort of easy to access that we could study. And it turns out the peritoneal cavity, where most of your organs are sitting, is sort of ticking all the boxes for this kind of study because it has a well-defined tractable immune ecosystem that has various immune cells belonging to both the innate and adaptive compartments like macrophages, B cells, T cells. It's also important in the general homeostasis of the cavity, but also in response to acute issues like peritonitis, so it has physiological relevance. And then finally, it's also very easy to access through postmortem peritoneal lavage. You don't need to do any tissue dissociation that can perturb cells and their gene expression and their function. You can just get the cells using a PBS-BSA injection in the peritoneal cavity postmortem. And so all of this gave us like a really nice way to study what's going on with age in an immune niche. And so we used aging mice. And so we have, in this case, in each experiment, only one provider, but we have both mice from the aging mouse colony at NIA and some aging JAX mice, and we see very similar results in both. So I won't specify which one came from which moving forward, but we get very similar results from both vendors. And so we use, for the young time point, we use 4 to 5 months of age. We do this because this is about when the hormonal cycles in young female mice are settled. So instead of looking at teenagers, we're actually looking at young adults, at least in that angle. And we compare them to old, 18 to 20 months of age. And we use that age frame because at least in the C57 black sex background, by 22, 24 months of age, about 80 to 90% of the mice have pituitary adenoma, which we're afraid might give us a false idea of what might be happening in hormonal axes. And so, as I said, we perform peritoneal lavage, and then sort of to unbiasedly figure out what's going on in that niche, we used single-cell RNA-seq using the 10x genomics platform. And so the good news is that we are able to find the cells we're supposed to find in that niche. We have a majority of B cells, and then we have a large population of macrophages, some monocytes, and a good number of T cells as well. And so not only we can identify what the cells are, but we can actually see which of those cell types, once we annotate them, are most responsive to the biological condition we're studying. So in this case, being young or old, female or male. And using a framework called OGRE, we can determine which of those cell type is most responsive transcriptionally. And what we found, and you can see here because that's the most intense red color, is that the macrophages in this case are the most responsive to female versus male, young versus old conditions, suggesting that they would be the most interesting for us to study. Now, we can also look at the relative abundance of those cells in the different libraries and see if there are changes in the composition. And we notice, and this has been observed by other people in either females or males, but never together, that we have a huge shift in the proportion of the cells in that niche, with an increase in the B cell fraction and a decrease in the macrophage fraction. And it looks like those changes are a little bit more extreme in the females than the males. For the T cells, we don't think this is real biologically because we've done flow cytometry and we were able to confirm B cells and macrophages, but not the T cells. And so at this point, the macrophages, we know they're changing in proportion, and we know they're changing at least at some level in their gene regulation. Now, single cell is very good for a lot of studies, but the problem is it suffers from a lack of sensitivity on the lower range of gene expression, just due to the way the technology works. And so since macrophages are very well-defined cell type, we could actually go back, purify them, and characterize them using bulk methods that are way more sensitive in that manner. And so to do this, we use the same type of mice. We do the peritoneal lavage. We purify the cells to get very high purity of F480 positive, CD11B positive, which those are those mature macrophages in the peritoneum, and then perform a multi-omic characterization of the cells for the transcriptome, and then their chromatin and epigenetic landscapes using ATAC-seq and cut and run for H3K4 trimethylation. And so what do we see? Well, what's really interesting, and so since those are very high dimensional data sets, we usually like to first project those in a 2D space to see what are the big differences, right, between the different samples. So this is multidimensional scaling. So what we observe, which is consistent with what I told you during the sort of the introduction of my talk, is that the biggest separation here in the transcriptome is actually whether the cells were purified from males or female animals. And there is a secondary sort of separation when you look between the darker colored and the lighter colored dots, suggesting there is a secondary axis for aging, but it's a little bit mixed. So it's definitely not as clear. Sex is definitely the biggest change in the transcriptome, and we observe genes that are significant with both variables. When we perform the ATAC-seq analysis, we see a very similar picture. If anything, the difference between the female and the males macrophage is even bigger. And same for the cut and run for H3K4 trimethylation, very clear separation between female and male samples, and secondary separation for aging. And so here it is very clear that at the basal state in young healthy animals, those cells are different. But in our lab, since we're interested in how, the difference of the trajectory, right, how they age, this does not tell you if there's a difference in trajectory. It just tells you there's a basal difference you're working with. And so in a very sort of naive way that we look at this is we say, okay, well, even if they start from different spaces, we can look at how they react to aging by looking at the rate of change in gene expression with age in the female macrophages here on the x-axis, compared to the rate of change in gene expression with age in the male macrophages here on the y-axis. And the idea is that if they go along very similar trajectories with aging, this should be very correlated. And so although there is a significant correlation, this is a Spearman rank correlation of 0.288, this isn't super impressive, I'm sure, to any of you. And so that suggests that there is not a great correlation. And so if we move a little bit further and we look for genes that were significantly changed with age only in the females or significantly changed with age only in the males, those are labeled in pink and blue dots. And you can see that there is definitely a nice population of genes we can identify, and no clear pattern of where they are in the graph, suggesting that they're mostly decorrelated with each other on what's going on with age on those genes, suggesting that the aging trajectory of those peritoneal macrophages are largely sex-specific. So, it's great. We have genes changing. We have peaks changing because we have a similar view when we look at the ATAC-seq and the cut and run. But really, we're biologists. We're curious about what that means biologically. And so we decided to perform enrichment for gene ontology terms in the female and the male macrophages with age, and then focused on the pathways or the terms that end up being significant only in one sex with age. And this here, I'm showing you only the top 10 in each sex. So here in that first column is the top 10 most significantly regulated pathways in female aging macrophages, and in blue, the top 10 pathways most significantly regulated in male aging macrophages. And we noticed a couple of things. Notably, here there's phagocytosis recognition pathway and some membrane pathways here, the endoplasmic reticulum, plasma membrane, suggesting that maybe something's going to be going on with phagocytosis, which is a key function of those cells. Now, the problem is gene ontology in general has a very poor annotation for phagocytosis regulators. So we also wanted to confirm this using better lists. And so for that, we took advantage of 2 CRISPR screens that were published in the past few years that identified over 500 new phagocytosis regulators in macrophages and macroglia. And using those and performing GSEA like we did in the previous analysis, just for those phagocytosis regulators, we see a very clear downregulation of those phagocytosis regulators in the female macrophages with age. And in the males, it's actually non-significant, but if anything, they're trending up. Suggesting something's going to happen to the phagocytic capacity of macrophages with age in females, but this will not be happening in males. So phagocytosis is something we can easily test. And so we're doing this using a fluorescent cargo, in this case, green labeled zymosan. We give purified macrophages about an hour, which allows macrophages that are competent for phagocytosis to internalize those bioparticles, and then we don't wait too long, because if we wait too long, those will be destroyed. And so that's about 1 hour. And so we do this by microscopy and flow cytometry. I'm going to show you the microscopy results here. And the idea is we're looking for the fraction of cells that internalize that green cargo. And so when we look at this, consistent with the omic analysis I've just shown you, we see that there's a significant decrease in the phagocytosis capacity of female macrophages with age, which we do not see in the males with age. If anything, the males are trending up, even though it is not significant, suggesting that there is a functional impact of aging in the females specifically. Now, if we go back to those GO terms and we look at other potentially interesting factors, we also noticed a lot of metabolism pathways that are regulated specifically, in this case, males, but not females. But a lot of metabolism pathways altogether were also regulated in a sex-specific fashion. And so you might be wondering why we care about this, because, macrophages. Well, it turns out that the metabolic preference of macrophages has been associated to their phenotype. So-called M1 macrophages, which is more of an in vitro designation, but let's say more pro-inflammatory macrophages tend to favor glycolysis over respiration. And more tissue repair, tissue remodeling macrophages tend to prioritize mitochondrial respiration over glycolysis. And so the metabolic preference of macrophages can tell us something about their function. And so we, again, use different techniques, but I'm showing you here what we've done using the Seahorse platform with the Cohen lab at USC to measure that glycolysis respiration preference. And the cleanest results were on the glycolysis side. So I'm showing it to you here, where we're measuring the extracellular acidification rate after injection of specific components. And we can deduce the glycolytic capacity using that second step. And so here I'm showing you the data from one cohort. We have 3 cohorts worth of data to show you the dynamics of it. But basically, we do see that in general, the young females in light pink have the lowest extracellular acidification rate of all the groups, suggesting that they have the lowest glycolytic preference. And indeed, when we sort of extract the glycolysis from 3 different cohorts and put it all together, we do see that the female macrophages tend to increase in their preference for glycolysis with age. And again, we don't really see this in the males. And so there is sort of a specific skew to become more glycolytic with age in the female macrophages, which also tends to be associated to a more inflammatory phenotype. And so anytime you see, sex-specific or sex-dimorphic phenotype, you're curious to see where this comes from, right? What is the mechanism driving these changes? And obviously, when you see sex differences at the basal level, those can come from difference in gonadal hormones, right? Females tend to have this estrogen bias, males tend to have the androgen bias. There's also the role of sex chromosome complements, being XX versus XY, and that's something that Ashley's talk earlier today addressed a little bit. But both of those can have, impact. Now, the only one of those that does change with age, because your karyotype would be largely similar. The thing that changes dynamically with age is actually the level of hormones. There is a decrease in estrogens that's quite dramatic, even though in mice it is not as dramatic as in humans. With age, androgens decrease slightly, not to the same level. But we were wondering if maybe the female-specific phenotypes might be due to the loss or actually dramatic decrease of estrogen systemically. And so I'm just going to show you 2 snippets of data. We think that it plays a role. And we basically looked at conditions where we can naturally or artificially reduce estrogens in young female mice and see what that leads to. The first one is just looking at naturally cycling mice, because as mice go through the estrous cycle, there are times in the cycle where they naturally have high estrogen levels and times in the cycle where they have naturally low estrogen levels. And so if we just take a population of mice on any given day, we're going to have mice in those different phases, and we can isolate their macrophages and look at the phenotypes. And indeed, what we can see is that when we compare high estrogen phases of the estrous cycle, so proestrus and estrus, to low estrogen phases of the estrus cycle, like metaestrus and diestrus, we do see a decrease in the phagocytic capacity of those cells. And to give you context, the difference in the relative estrogen levels between those high and low estrogen phases of the estrous cycle is going to be similar in amplitude to what's going on between average young female and average old female. Now, if we do this sort of artificially by performing ovariectomy or sham surgery on the mice and then waiting a month and getting their macrophages, we can also see that removing the gonad, which will altogether remove the bulk of estrogen synthesis, also leads to decreased phagocytosis. And so we do think that the fluctuations in estrogens can promote decreased macrophage phagocytic capacity. I don't have the time to show you from the metabolism, but the metabolism data suggests that the metabolism is not regulated by estrogen loss, or at least not fully. And so we've been curious in seeing if we could use all that omic data we generated to identify potential drivers of those phenotypes outside of estrogen signaling. And so basically, what we've been doing is looking at things that happen only in the female macrophages with age and not in the male. So first, which transcription factor genes are specifically regulated in the females with age? And so we have a number of those, and I'm going to pull your eye to IRF2, which is high in the young female macrophages, but low in the older ones. And it's a little bit all over the place for the males. Next, we can look for in the peaks, the regions that are accessible by ATAC-seq, or the peaks that are marked by, sorry about H4, it's H3K4 trimethylation by cut and run, and look at the peaks that are specifically remodeled with age only in the female macrophages, but not the male macrophages, and look for motif enrichment. Here, I'm just going to show you the ATAC-seq, but we have similar data on the cut and run. And again, one of the top enriched motifs, specifically in the peaks that are remodeled only in the females, is IRF2. And then when we go into our single-cell RNA-seq data and we look for transcription factor regulon activity change with age using a package called CNIC, we also see a specific decrease in the IRF2 regulon in the female macrophages with age. And there is a small decrease, but it's not nearly the same amplitude in the male macrophages. And so this tells us IRF2 might be playing a role here. And so right now, Ryan is optimizing knocking down IRF2, overexpressing IRF2 to see if we can mimic or rescue the aging phenotypes. But we are pretty optimistic because there is this data set that was generated in bone marrow-derived macrophages from wild-type and IRF2 knockout mice that we've reanalyzed. And the phagocytosis regulators that I mentioned earlier when we analyzed our own RNA-seq seem to be downregulated significantly in the knockout IRF2 macrophages. And then there was another study that looked at the metabolic effect of IRF2, and in that study upon siRNA to IRF2, there's an increase in glycolysis, similar to what we see with aging. And so we believe that this could be sort of a good lead to follow right now and understand exactly what's going on specifically in those female macrophages. And so with that, I've shown you that we have good data that resident macrophages age in a sex-dimorphic manner in mice, that you can use the omics remodeling to predict what's happening functionally to cell populations, that the age-related loss in estrogens could drive aspects of the female-specific functional decline, although not all of it, and that we're now following up on IRF2 as a candidate mediator for some of those other phenotypes that we've seen with macrophage aging. And so before closing, I always like to sort of have a PSA where people keep saying, we want to have personalized medicine, personalized therapies, with the idea that, if you have this SNP or that SNP, we can give you this drug or that drug to improve your chances of responding. But, the biggest genetic differences of them all is a whole chromosome. And so I think it would be very important as a field and, as scientists, to start thinking of sex as the first level of personalization of therapies, and maybe start personalized medicine by taking into account whether the patients we're trying to do something for are female or male. And so with that, I'd like to thank my fantastic team, all super talented people who've trusted me and work in the lab. I want to highlight Ryan here, fantastic grad student who's spearheaded most of what you've seen today. This work was obviously would not be possible without our funders, specifically here the Pew Foundation. And I want to thank our fantastic collaborators, specifically Helen Goodridge, who's been a fantastic resource on the biology of macrophages and the innate immune system. And I would be happy to take any questions. Thank you.
All right, great, Berenice. Really nice talk. Super exciting. All right, so there's a question that came in on the Q&A, and I'm curious about this too. It's related to so the person who submitted the question is Amin Hagani, and it's related to the use of mice versus humans, given that mice are not, in our mouse facilities, are not generally exposed to pathogens. And so could you comment on the use of mice that are not exposed versus humans in these types of studies?
Yeah, I mean, obviously, the end goal is to look at humans. Unfortunately, it is not ethically acceptable to do human knockouts. So for mechanistic studies, we need to use models. And so there is actually some fantastic work being done at NYU, where people are rewilding mice to force them to acquire, wild microbiome, and then bringing them back to study them and what happens. And there are big differences. And I agree, that would be really interesting to look at, but we have to start somewhere. And so even though there are limitations, I hope we can learn something important from looking at the mice.
Great. So I have lots of questions. So one question I have, and sorry if you tried this and I missed it, but have you boosted estrogen levels and looked at some of these phenotypes in the old animals?
Yeah, so we've tried this, and the short answer is we don't get any rescue. Pretty much we get partial rescue on one phenotype that I didn't talk too much about, but we don't see rescue. But one of the things that is not, very well appreciated by the community is that the ability to respond to estrogen is highly regulated by estrogen. And so the relative level of the 3 estrogen receptors and how they're expressed is affected by estrogen. And the way you respond to estrogen depends on the relative expression of those. And the expression of estrogen receptor alpha is actually one of the top most regulated genes that we see in response to aging in the female macrophages. So we think the only way you could potentially fix it, but this is long-term, so that might be in our future, would be to basically start supplementing just as they enter oestropause. So we would have to start supplementing them when they're 14 to 15 months and then follow them to 20 months. But it's difficult to get approval to give sex steroids to mice for that long because the IACUC's afraid we're going to give them cancer. One of the things we're trying to see is if we can do adoptive transfers. So take the old macrophages and transfer them to young mice to see if we can fix things. And hopefully that can partially answer that question. But yeah, it doesn't look like short-term supplementation in old mouse can fix the problem.
Great, thanks. So yes, I guess it's not that simple, unfortunately. Okay, so a few more questions coming in that I want to get to you from the Q&A. Okay, so one question on the activation of gene expression in males versus females. So is there any basic activation which happened early on in macrophages in the females versus the males? Like, I think activation of the macrophages themselves.
Does it happen at a different rate? So the short answer is I don't know. I don't think it's been looked at. Now in younger animals, like 2-month-ish, there's been some work showing that there is a different rate of self-renewal of those macrophages because they are mitotically competent. There's a different rate of self-renewal in the females and the males. At the age we're looking at, it doesn't look like self-renewal is much of an impact, but we're still looking into it. So I will have a full answer at some point in the future.
All right. So another question from Yusra Ajuao. Great talk. Did you look at other histone modifications? Do you think that epigenetic modifications might be sex-dependent or sex-specific?
That's a great question. So one of the issues is that when you're looking at naive macrophages from the peritoneal cavity, and I know that's going to make you laugh, Ashley, because it's plenty compared to neural stem cells, but we only get 600,000, 700,000 cells per mouse, at least in the young animals, and we're limited by that, which means that already doing everything we've done is a lot of animals. So we haven't gone to other marks. To answer of whether I think the epigenome in general is going to be sex-dimorphic, yes, I believe so, if only because sex hormone receptors are chromatin remodelers. They can kick off nucleosomes and remodel chromatin around them. And also because escapers on the so-called inactivated X include things like KDM6A, which is a histone demethylase, and being expressed at double dose in female cells versus male cells would definitely have impact. So yes, there will be huge differences in the epigenome in general, and I do believe they will be impacted by aging.
Great. So Alessandro Bonetti wants to know whether you considered using nascent RNA-seq instead of the global transcriptome to further pinpoint differences in transcriptional response and efficiency between the sexes.
So again, great question. And I believe that would be really interesting to look at, but nascent RNA-seq work also requires more cells than we can spare. So we would have to look at another niche where there would be more cells.
And second question from the same person, whether you've considered looking at whether female genomes tend to accumulate more double-stranded breaks compared to males.
Excellent questions. We don't know, and we haven't looked yet, but you know, there's so much. If people are interested in double-stranded breaks, I just want more people to look at sex differences. So please look at it.
And another question sort of related to this this thread, I think you already mostly answered this, whether you've looked at other epigenetic modifications like DNA methylation. Have you looked at that in the different sexes?
We have not, but I know from some publications that people have seen big differences in the DNA methylation of females and males. And actually work from Morgane Levine, who's going to be talking in a couple of days, showed that there is an acceleration of the DNA methylation clock after menopause. So definitely it's very likely there would be differences, but we haven't looked ourselves there.
Great. And then this is from an anonymous person. Is there a senescence deepening that's different between males and females that you know of?
Okay, so this is where we haven't looked at cell senescence itself. I know that I've had some, coffee conversation at conferences with people who've working on senescence, who've told me that the rate at which cells enter senescence and the actual SASP phenotypes are different between genetically male and female cells. So there is likely to be differences, but we haven't looked ourselves, so I can't give any details there.
Yeah, great. And then just more generally in terms of the immune system, are the immune cells functioning differently between males and females?
Yeah, it does look in general, and obviously every time you make a general conclusion, there's probably exceptions somewhere, but it does look that in general, the female immune system is more active and more aggressive in the adaptive branch, but a little bit less, at least in young, healthy females, less in the innate branch, whereas the male immune system tends to be more active on the innate branch and less on the adaptive branch.
Interesting. Okay, a little bit more on the sex-specific macrophage phenotypes. So can the differences observed here be a reflection of an expedited aging process in females, meaning that they will eventually be observed in a much older male?
So that's a very interesting comment. So I think it is possible. And also because one thing that is not necessarily well appreciated is that males actually have quite high levels of estrogens from aromatization of testosterone. So, as testosterone levels go down, you would also have decreased estrogen in males. So anything that's driven by decreased estrogen could also happen eventually in the males. And people who've looked at the phagocytic capacity of macrophages in much older animals, generally in males, do see a decrease. So it is possible that what we're seeing is accelerated aging in the females. Whereas the molecular networks behind it are going to be the same or not, I don't know, but that's an interesting thought.
Yeah.
Ashley, I think if you could, Gary has his hand up. If maybe he can open his mic and ask his question.
Sure. Maybe not. Do you want to try again? You can also put it in the chat. Okay.
Yeah, maybe we should just go with
Can it work now?
No, still an echo. I can go ahead and ask. He says, maybe he missed this, but do you think why do you think there's an increase in preference for glycolysis with age? Are age-related effects of mitochondria activity involved, do you think?
So for this, I didn't show the mitochondria data because basically nothing's happening. So we think the mitochondria, at least at the ages we're looking at, are fine and they function fine. It's just really that the cells are shifting to a glycolytic preference. And this is consistent with that M1 phenotype I mentioned, where the macrophages become more pro-inflammatory and start preferring glycolysis. We actually also notice, so there are people who've looked at when you treat macrophages with LPS, so you make them more M1, some genes are upregulated or downregulated. And so when we look at those genes in our RNA-seq, the female macrophages are very clearly shifting to a more M1 phenotype and not an M2 phenotype, whereas the male macrophages with age don't have that shift. And so it's likely that the female macrophages are just experiencing a more inflammatory state with age, and the males are not, and that would be the reason they shift their metabolism, not because mitochondria becomes effective.
That's interesting. There's one more in the Q&A. Do you think that the sex differences in macrophages might be tissue specific? Like if you look in different places?
So this is actually a project that we have a fantastic computational undergrad student looking at using publicly available data and data we've generated. And I can tell you, yes, they're going to be extremely tissue specific. The aging part, not as clear because that data does not exist, but the sex differences at basal are going to be very tissue specific for macrophages.
All right. Great. So do we want to move on to the panel? That was fantastic. Thanks for the great talk and discussion. Really.
Thanks, Ashley. So yeah.